Good morning, good afternoon, everyone. Thank you for attending our presentation. The title of our vaccine design is Rabies VLP Vaccine, Expression and Purification of Surface Antigen G protein of rabies virus-like particles in Pichia pastoris. Our group members are Iago, Luciana, and Poojah. This is our table of contents, which consists of the introduction, study objectives or goals, materials and methods, the expected results and discussion, and finally, conclusion and final remarks. This is the introduction. Rabies is a vaccine-preventable zoonotic viral disease. It is characterized by an acute, progressive, fatal inflammation of the brain and spinal cord. Once clinical symptoms appear, rabies is virtually 100% fatal. All mammals are susceptible to rabies, and the principal reservoirs of this virus are of the order Carnivora, which are dogs, cats, foxes, raccoons, and skunks, the order Chiroptera, which consists of bats, and other reservoirs include domestic and wild animals. In 99% of cases, domestic dogs are responsible for rabies virus transmission to humans. Approximately 60,000 deaths each year globally occur in about 150 countries. Globally, rabies causes an estimated cost of $8.6 billion per year. Rabies actually originated from the Sanskrit word rabas, which means to do violence, or the Latin word rabere, to rage. The Greeks also called rabies lysa, meaning frenzy or madness. The rabies virus belongs to the order Mononegavirales family Rhabdoviridae and genus Lysavirus, and the species is Rabies lysavirus. And according to current taxonomic classification based on phylogenetic analysis, the rabies virus belongs to phylogroup 1. The rabies virus is found globally in different countries and has been isolated in different countries. And as I've mentioned earlier, the primary reservoirs are all mammals. This is the continental distribution of lysavirus reservoirs. So in Brazil, dogs and crab-eating fox, as well as vampire bats, are reservoirs. And in Africa, including South Africa, dogs, bat-eared foxes, as well as the yellow mongoose are the primary reservoirs. It should be noted that in Australia, there is no terrestrial carnivore reservoirs for rabies virus. Brazil has one of the largest estimated populations of dogs worldwide, around 30 to 50 million dogs. According to the Brazilian Health Information System, SINAN, dating from records from 2008 to 2017, an average of 506,148 bite injury patients per year occurred, equivalent to an incidence of 255 bite injuries per 100,000 people per year, which is quite a lot. And the rabies vectors in Brazil, especially in Sao Paulo and Rio Grande do Sul, are vampire bats, insectivorous bats, capuchin monkeys, and crab-eating foxes. In the northeast region of Brazil, the marmoset is the vector, and during 1998 to 2016 in Sao Paulo, an average of approximately 3 cases of rabies per year occurred, and most of them were transmitted by bats to dogs and cats and can be classified as enzootic. Only 52 cases have been diagnosed in the past 18 years. Bats are also reservoirs for emerging rabies virus diseases. Rabies is a notifiable disease in South Africa, especially in the KwaZulu-Natal, Eastern Cape and Limpopo provinces, according to the Health Act from 1977, whereby 92% of cases are due to rabid dog bites, and mostly children under the age of 10 are affected, as shown in the photograph. Approximately 600 to 700 cases of rabies occurs annually in South Africa, which have been diagnosed in domestic and wild animals, and the rabies vectors include dogs, cattle, and the yellow mongoose, which account for 85% of all confirmed cases. The black-backed jackal and the bat-eared fox are other vectors, and there is a problem with regards to dog rabies epidemics in parts of South Africa since 1987, which originated from the southern Mozambique dog endemic regions, and a total of 834 cases of animal rabies were laboratory confirmed in 2012, which 508 cases were related to dogs, 212 cases were related to domestic animals, and 114 cases were related to wildlife. This is the global distribution of dog-transmitted human rabies, showing the global map, whereby regions in yellow show that rabies is absent, light green regions show that rabies is suspected, and dark green regions show confirmed cases of dog-transmitted human rabies, and the light blue areas are unknown cases of dog-transmitted human rabies. In figure A, as you can see, bats are the primary reservoirs, or I would call them the primary hosts. They seem to be immune to the rabies virus, and they carry both current and emerging rabies viruses, and they can infect humans directly, or they can infect dogs and other mammals. And when these secondary hosts, which are dogs and other mammals, domestic animals, wild animals, when they bite humans, they spread the virus. And if not treated in time, and once symptoms develop, the disease is fatal or deadly.
So moving on to the viral genome and its structure, the first picture shows rabies virus genome and how it is organized. The protein sequences are in the following order: N, P, G, M or M2, and L. Each sequence is spaced by a non-coding intergenic sequence. Its genome is composed of a single-stranded negative sense RNA, and it's about 12 kilobases. In regards to the structure, rabies virus is a bullet-shaped particle in which one end is conical and the other is flat. The genome encodes 5 proteins: an RNA-dependent RNA polymerase, or L protein, which is represented by the red circles. Next, there is the nucleoprotein N in blue, which binds to the viral genome and forms the nucleocapsid. The phosphoprotein P in orange is associated to the polymerase and assists in the replication and transcription activities. The viral nucleocapsid is enwrapped in a lipid bilayer derived from the host cell, in which matrix protein M2 and glycoprotein G are associated to this membrane, forming the viral envelope. These 2 proteins play a vital role in viral cycle and pathogenesis. G protein mediates fusion of the viral envelope and cellular membrane, and both G and M2 proteins are responsible for virion formation and particle budding. Now in more detail about the protein we have chosen to use as an antigen for this project. The G protein is an integral transmembrane protein with 505 amino acids. It forms trimers in the viral envelope, and it is the major antigen responsible for the induction of immune response. It has been shown that the G protein is able to activate T helper cells, cytotoxic cells, as well as eliciting production of neutralizing antibodies. So this antigen can activate both humoral and cellular response, which is an interesting target for vaccine design. Another feature of these proteins is the autonomous exocytosis activity. That is, the G protein can initiate vesicle formation by itself without the need to interact with another protein. In fact, G protein has been found in infectious particles in the supernatant of infected mammalian cells, demonstrating how important this protein is for the proper formation of viral particles. So in this slide, we show a real representation of rabies viral particle by electron microscopy. It is possible to visualize the viral particle shape and also the envelope. So about the virus cycle, rabies virus entry is mediated by G protein, which is responsible for receptor recognition and fusion of the viral envelope to the host cell membrane. The virus enters the cell by endocytosis and it releases its RNA genome inside the cell. So this negative sense RNA is used for replication and making of new RNA copies and for transcription as well to produce new viral proteins that will form new viral particles. For instance, proteins M2 and G are transferred to the cell membrane, and proteins P, L, and N will associate with the genome to form the ribonucleoprotein. M2 and G can also interact with N protein to assemble and budding out new viral particles of the host cell. The pathogenesis of the rabies virus is represented in the right picture. The virus is inoculated into a host by an animal bite. First, it replicates in the muscle tissue until it reaches and crosses the neuromuscular junctions. This is where it gains access to the nervous system. The virus travels via axonal transport to the nervous system, where it replicates in the motor spinal cords and ganglia. Once the virus infects the brain, it causes a fatal inflammation or encephalitis. The last step in the cycle is the replication of the virus in the salivary glands, so it could be retransmitted to another host. So moving on the transmission aspects of the virus, transmission of rabies could occur through contact between infectious saliva with broken skin or mucous membranes, bites, scratches, or leaks. Also, it could be transmitted through consumption of carcasses or infected animals. Abrasions in the oral cavity could allow the passage of the virus into innervated muscle and neuronal tissue. The virus is maintained in most parts of the world via mesocarnivores, which are animals whose diet consists almost exclusively of meat. Some of them are dogs, foxes, raccoons, mongooses, and skunks. There are a few exceptions, such as multiple bat species, kudu, and cattle, which are herbivores. But it has been shown that they can serve as reservoirs for the virus as well. In the case of humans, cattle, and equids being infected, those cases are called as spillovers, which can occur when the virus gains the ability to infect a member of a new host species. The virus causes a variety of symptoms in infected animals. So rabies could cause neurological diseases in mammals, and once the clinical signs develop, it is usually fatal. Symptoms such as stop eating and drinking, withdrawal, lethargia, or fever, vomiting, and anorexia could show. Incubation period could vary. It could depend on a lot of factors. For instance, the amount of virus transmitted, the virus strain, the host immunity, nature, and site of the wound also could affect the incubation period. In dogs and cats, the incubation could vary from 10 days to 6 months. The disease could progress in two different forms. The first one is the paralytic form. It could cause paralysis in the animal, so the animal might have difficulty to swallow and salivate profusely. Also, facial paralysis, confusion, apathy, lower jaw drop, and also tachycardia. The second one is known as the furious form, which represents an infection of the limbic system in the brain. It could present as a hypersalivation state, also hydrophobia, hyperactivity, and hallucination are all possible symptoms.
Vaccines for rabies are used worldwide. In this slide, we can see some rabies vaccines used. All the vaccines are inactivated and have the hydroxyaluminum as an adjuvant. In Brazil, we have commercial vaccines for rabies. However, the government also encouraged the population to vaccinate the domestic cats and dogs by providing a vaccine campaign against rabies. Many side effects are reported after the campaign vaccines due to a preservative used in its composition, the thimerosal. Inactivated vaccines can lack in immunostimulation, being essential and adjuvant in the composition. There are some problems with the current vaccines for rabies. The cost of production due to high expense of cell lines is one of the problems faced. Also, maintenance and storage are a problem due to low temperature needed to preserve the vaccines. Low-income countries suffer with the limitations in the transportation, availability, and accessibility of vaccines. Additionally, narrow vaccines may be a risk of rabies. Inactivated rabies vaccines may be activated in some specific hosts. Some rabies vaccines are still animal-derived. Purified chicken and duck embryo are still in use at the vaccine production. VLPs are formed by structural viral proteins, which have an inherent property for self-assembly and mimic the morphology of the pathogen. In contrast to live viruses, VLPs are non-infective and non-replicating, since they are essentially devoid of infectious genetic material. VLPs displays antigenetic epitopes in the correct conformation and in a highly repetitive manner, leading to a cross-linking of B-cell immunoglobulin receptors and B-cell activation. As exogenous antigens, VLPs are efficiently taken up by professional antigens-presenting cells, particularly dendritic cells, followed by antigen processing and presentation by MHC class II molecules, the C activation, and maturation through upregulation of co-stimulatory molecules and cytokine production, and stimulation of CD4 T helper cells, leading to the induction of a strong humoral and cellular immune response. Similar to native viruses, VLPs are processed in the cytosol of disease and are presented by MHC class I molecules to cytotoxic CD8 T cells by the cross-presentation mechanism, inducing potent cytotoxic immune response. Study objective goals. The main aim of our study is to construct, express, purify, and characterize rabies virus-like particles, known as VLPs, in Pichia pastoris. The study objectives are: construct a recombinant yeast VLP that expresses the G protein and evaluate the particle production. Immunize mice with the VLPs and evaluate the immune response triggered. Evaluation of alum adjuvant that can be used with the yeast VLP expressing G protein to give the best immunogenic response in mice.
We will move now on to describe the material and method section of our project. So for the first part of our project, it would be the G protein expression. So we have chosen yeast as the expression system, specifically Pichia pastoris. Pichia pastoris is a kind of yeast that has been largely used as an efficient protein expression platform. And also, it has been used for VLP production. So it's a safe platform for protein and VLP expression, and cheaper than mammalian cell culture or insect cell culture. So that's why we have chosen it. Although it is capable of adding post-translational modifications to their expressed proteins, the glycosylation pattern differs from mammalian cells. So this could be a disadvantage in terms of immunogenicity, but we would evaluate if this glycosylation pattern would make an impact on the final result. So the first step would be to identify the G protein sequence, and we did that by accessing NCBI and using as a database to get the full sequence for a protein. Once we have our sequence of interest, we can amplify it by PCR and insert it into the plasmid vector. And we have chosen this one because it has already been used as an expression vector. It contains a multiple cloning site with several restriction sites that could be used to insert or sequence by restriction enzymes. Also, it contains a selection marker, which is the antibiotic zeosin, so we can select the transformants later on. And lastly, it adds a polyhistidine tag to our protein, which allows us to use metal affinity chromatography as a purification method. So continuing the cloning process, so yeast cells would be transformed by electroporation. It is a commonly used process for plasmid entry in yeast cells, and the transformer will be cultured in a medium containing the antibiotic zeosin, so only the cells who have the plasmid would survive. And now for the G protein expression, so the single colony transformants will be harvested and cultured in a liquid culture medium, and expression of G protein will be induced by adding methanol to the medium. So our chosen plasmid also contains an expression promoter that is controlled by methanol. So for our protein to be expressed, it would be necessary to have methanol in the medium. So that's why we added. So by the end of this part, we would expect to have our protein secreted and forming VLPs, but we know that this could also happen later. So we will collect and centrifuge and work with the supernatant. So to confirm the G protein expression, we will use electrophoresis to visualize if our sample contains proteins in this expected molecular size. And to confirm that is indeed the G protein, we will perform Western blot using specific antibodies that will bind only to the G protein. The next step would be the purification step. So for the purification method, we proposed methyl affinity chromatography. Since our protein contains a polyhistidine tag, this tag will interact with nickel resin and be separated from the rest of the proteins that our sample may contain. So we will obtain a purified fraction of the G protein, hopefully in a VLP form. And in order to confirm the formation of VLPs, we will analyze this purified sample via electron microscopy. So using this technique, we will be able to visualize and measure the particle size. And the last part in the characterization of our G protein VLPs will be the quantification step. So in this step, we will be quantifying the protein content of our samples in order to establish specific doses for our future tests. This will be accomplished by the BCA assay, which is a very sensitive method that is largely employed in protein quantification.
For the immunization plan and the preclinical tests, mice will be used as animal model. The immunization plan will last 21 days, and 3 immunizations will be conducted in days 1, 7, and 14. The animals in the study will be divided in 4 groups. The first group will receive only PBS. It is the control group. The second, third, and fourth group will receive low, medium, and high doses respectively. To evaluate the alum adjuvant at the preclinical tests, mice will be used as animal model. The evaluation will last 21 days, and 3 immunizations will be conducted in days 1, 7, and 14. The animals in the study will be divided in 4 groups. The first group will receive only PBS plus alum. It is the control group. The second, third, and fourth group will receive low, medium, and high doses with the adjuvant, respectively. For the immunization plan, on day 0, blood will be collected. On day 1, the animals will receive the first dose. On day 3, blood will be collected again. On days 7 and 14, the animals will receive the second and third boost respectively. Finally, the animals will be euthanized and the blood will be collected on day 21. The immune response will be evaluated by analysis of the IgG titer by ELISA, analysis of neutralizing antibodies by PRNT, analysis of interferon gamma by ELISA, and cell proliferation assay will be conducted. For the challenge test, the animals will be challenged with RBV 2 weeks after the last immunization. All animals will be confirmed to be seronegative for rabies virus using serologic assay. Groups of 5 mice will be intramuscularly vaccinated with 3 doses of VLP rabies vaccine containing low, medium, and high doses. Also, the control group will be injected with sterile PBS. The animals will be analyzed based on the clinical signs. Furthermore, an analysis of the viral load in brain and blood by viral titration or real-time PCR will be conducted. For the survival test, the same methodology used in the challenge test will be performed. At the end, the survival percentage will be measured.
Expected results and discussion. These are some of the expected results of the vaccine design that we can expect based on some articles that we found. Okay, so in this slide, the figures were taken from an article by Gupta et al. They used G protein as an antigen, and this was encoded in the ORF2 region, and they used this to create a transformant Pichia containing a PPIC Z alpha A ORF2. Figure A is a schematic of the mice immunization schedule to evaluate the immunogenic potential of the VLPs, as you can see. An immunization assay was performed in 6-week-old mice, and they were injected with the VLPs as shown in this figure. So on day 1, pre-immunized blood was collected and the immunization was performed. On day 15, blood was collected and the first booster injection was given. On day 29, blood was collected and the second booster injection was given. And on day 43, there was euthanization of the mice, followed by collection of blood and splenocytes. So at the bottom, you can see the different groups that were involved in the study. In group 1, which is the control group, they used PBS to inject the mice. In group 2, they used alum. In group 3, they used the VLPs at different dosages at 1 microgram, 3 micrograms, and 5 micrograms respectively. In group 4, they used the VLP and together with the adjuvant alum at a dosage of 1 microgram, 3 micrograms, and 5 micrograms respectively. So why did the authors and why did we decide to use alum adjuvant with the vaccine? The idea is that by adding alum along with the vaccine antigen itself, the inoculation mimics a natural infection, and by doing so, it stimulates the immune defenses, such as the dendritic cells, to trigger a stronger immune response than the antigen alone, so making the patient or the individual immune to a real infection at a later date. In figure C, the cellular immune response elicited by the RV-VLPs was done by splenocyte proliferation assay. The splenocytes collected from 5 microgram VLPs, 3 microgram VLP plus alum, and 5 microgram VLP plus alum immunized mice could be significantly induced to proliferate compared to the controls, as you can see in the figure of PBS and PBS plus alum. For 5 microgram VLP plus alum groups, they were significantly higher, as you can see in the figure, as compared to only the VLP. This figure shows the expected transmission electron microscopy results following protein purification of the ORF2 112 to 608 amino acid protein. The protein was found to form VLP structures, as you can see, which could be observed using transmission electron microscopy. And although the population obtained was heterogeneous, a large proportion of particles were of 22 nanometers in diameter and were visible. And this suggests that the purified ORF protein encoding the G protein antigen assembled into VLPs, as you can see. In this figure, we have the analysis of viral loads in tropism organs. So the lung virus titers of whole inactivated hemagglutinin A immunized mice were infected with influenza H1N1 strain was performed, and on day 3, 5, and 7 days post-infection, lung tissues were collected and the viral titers were calculated using embryonated chicken eggs, and the results were expressed as log 10 EID 50 per gram of tissue, and the limit of virus detection was set at less than 0.1 log 10 EID 50 per grams. So what is the EID 50? It's when one EID 50 unit is the amount of virus that will infect 50% of inoculated eggs, and EID stands for embryo infectious dose. So in the case of our study, we would probably collect brain samples from the mice of our animal or preclinical model, because rabies infects the spinal cord and brain tissues. Coming back to the bar chart, as you can see, in the control group, the highest lung viral titer was observed at 3 days post-infection and persisted until 7 days post-infection. However, viral titers peaked at 3 days post-infection in all immunized groups and decreased at 5 days post-infection before becoming undetectable at 7 days post-infection. The decreased lung viral titers of each vaccine group tended to be proportional to antigen amount in the vaccine, and these results confirm that 2 doses of the non-adjuvanted split vaccine provides protection to mice against the CA04 challenge.
Another type of expected result would be the quantification of neutralizing antibody titers. So we would analyze sera collected from mice to quantify it. This is the kind of result we would expect by performing a plaque reduction neutralization test, or PRNT. In this assay, the serum is incubated with the virus and formation of plaques are observed. So the higher the neutralizing antibody titer is, the fewer plaques would be formed. Vaccinated animals are expected to produce more neutralizing antibodies than control group. In this picture specifically, they compare different vaccine formulations, one containing inactivated virus and another containing VLP. And the formula with the VLP showed better results in terms of neutralizing response. Moving on to analysis of cytokine profile. This will be accessed by ELISA or ELISpot. Spleen will be collected from vaccinated and control groups, and splenocytes will be cultured in plates coated with interferon gamma capture antibodies. We would expect to see an increase of the cytokine concentration compared to the control group, since this indicates an activation of cellular response. Total IgG titers would also be monitored throughout the vaccination period, so we can evaluate the efficiency of booster immunizations. Testing different dosage of VLPs could have an impact on the production of IgG, so this type of result could help us to determine the best dosage for our vaccine formulation. Our last expected result analyzes the survival rate of mice after a lethal challenge. In this figure, they specifically tested anti-VLP immune sera in infected neonatal mice. They observed that all animals survived after treatment with the sera. In our experiments, we would test for a different goal, but we would still hope to see the same type of graphic, in which vaccinated group has a better survival rate compared to the control group.
Conclusion and final remarks. To conclude, we will immunize mice with VLPs preparations. The collected blood samples will be analyzed by ELISA and real-time PCR. The results that we expected to obtain should indicate that VLP will be immunogenic and capable of inducing both humoral and cellular response. For further studies, we pretend to evaluate the M and G protein co-expression. Also, evaluate the thermostability of the recombinant VLP vaccine and the generation of P. pastoris strain with the constitutive G protein expression. We would like to thank you all for watching our project, and for any doubts, we are ready to elucidate.